Good morning, good afternoon, or good evening for day 3. Depending on wherever you are in the world, welcome to today's QMed Focus on Fundamentals webinar concerning guidance for changes in a medical device, sponsored by Nelson Labs and broadcast by Informa. It's day 3, and we'll be discussing biocompatibility and biosafety. I'm Bob Sperber, and I'll be your moderator today. We have just a few announcements before we begin. This webinar is designed to be interactive. The dock of widgets at the bottom of your screen will allow you to learn about today's speakers, download resources, share this webinar, and participate in the Q&A session that takes place at the end of the presentation. The slides will advance automatically throughout the event. Lastly, if you're experiencing any technical problems, please click the Help widget found at the bottom of your screen. Or you can type your issue into the Q&A area, and we'll be glad to offer one-on-one assistance. Now on to the day 3 presentation, Biocompatibility and Biosafety, Evaluating the Impact of a Change. Discussing today's topic is Matthew Jorgensen, Senior E&L Expert at Nelson Labs. To learn more about Matt, please visit the Bios widget. And now, Matt, over to you.
Thanks for the introduction. Super happy to be here talking about something that I'm really passionate and excited about. When we talk about the types of challenges facing medical device manufacturers today and how that impacts the way their devices get to the market and how they interface with global regulators, changes and how that relates to biocompatibility is way, way high up on that list. There's supply chain disruptions everywhere, and that is layered on top of all of the regular changes that manufacturers make to medical devices anyways. So this is a big topic. It's one that can be really useful. So approaching changes and biocompatibility skillfully to know when you need to, let's say, redo an entire suite of biocompatibility testing or just write a couple paragraphs. This can be really powerful down the line as we all approach these types of changes. And it's one of the things I would say occupies maybe 80% of my time as an expert is just looking at changes and helping folks navigate those. So it's a great topic. I'm super excited to go through this and some examples. The way that I'm going to do this is I want to start with a really strong base. When we talk about changes in biocompatibility, like what angles on biocompatibility do we really mean? And what tools or ideas do we have to evaluate biocompatibility in a general sense? And this base really informs the strategy that we're going to use as we approach changes. So we'll start with a strong base, and then I'll go into just some examples of changes and how we've evaluated them in the past, or how we could evaluate them if it came across our desk. And at this point, I really want to pause, and I know it's a little bit, I guess, less typical for a webinar format, but if there's any specific types of changes that the audience would like me to discuss, I'm happy to go through whatever is most interesting for you guys, the listener. So in about, 10 slides or so, I'll go into the case studies portion of this presentation. But before I present any of the case studies that I've, more or less picked randomly out of a hat, if you would like me to talk about a certain type of change or even a specific change, I'd be happy to do that first. So if you have an idea like that, or if you have a specific type of change in mind, you can enter that as a question into the question and answer box on your screen. And I have somebody's filtering through that will group those and send them over to me, and I'd be happy to talk about those first before I talk about the catheter, the 3D printed thing, and the neurological sensor. Again, that I just sort of picked randomly because I think that they're interesting. I'd love to talk about yours first. Okay, so just to get into this, first I'll just a little bit of a brief personal introduction that expands on the introduction that we had a second ago. So my background is really academic, strong training in physical chemistry. It's more on the physics side. Taught university for a while, and spent a good chunk of my life in academia just doing pure theoretical research, especially with respect to how light interacts with matter and things like that, and a lot of analytical chemistry as part of that. However, I left academics about 7 years ago, went full on into medical device, been with Nielsen Labs this time, where I focus on leveraging all of that theoretical knowledge to a much more practical situation, evaluating the biocompatibility of medical devices and also from a toxicological perspective, study toxicology in the meantime, and now a board certified toxicologist as well. My one true claim to fame, though, is that I helped to create a flavor of Mountain Dew that you see on your screen there. That's Mountain Dew Whiteout. It's a lot more like smooth riding Mountain Dew, one that you might have as you sit and relax on a patio on the end of a summer day, rather than right before you do like a double backflip on a BMX bike. So that's my claim to fame. For that reason, a lot of folks call me Dr. Dew, and I welcome everybody here to call me Dr. Dew as well, as it's too late in my life to abandon that nickname. All right, let's get right into this. So biocompatibility, what is it? Why do we care in the first place? I told you I was going to start with a firm base, and this is really where we're at. So I like to revisit this and think about it from time to time. Our whole goal as medical device manufacturers and really doctors, so everybody in the field of medicine and CROs too, is that we really want to improve the quality of life for people, right? All of the patients out there that our devices touch, we want to improve their life. And to do that, we need to first ensure that whatever way that we're treating patients isn't going to harm them, right? So we need to be sure that we're not unintentionally causing harm to people that we intend to help. And if you look back through the history of medicine, and especially with medical devices, you can find examples, lots of examples actually, of ways that medical devices have irreparably harmed people in ways that we've learned from and now want to avoid, right? And so this thing, there's a deep personal and ethical mission here for all of us. We have to keep patient safety first and foremost. We are all committed to not producing anything and bring anything to the market that might inadvertently harm somebody. And for that reason, that's why as we approach a change, we need to ask ourselves, is there any way that this could harm somebody? Is there any way that this could harm somebody in a way that we should test to make sure that that's not going to happen? And so for medical devices, based off of experience and through conversation with global regulators and all that stuff, we've really ironed out the main ways in which a device could harm people. And when we approach a change, we're asking, okay, could this change produce any of these adverse effects or biological endpoints that might produce harm? And those are all those things listed on the top of Table A1 and 10993-1. So I show these here. There's a long list of them. We'll talk about them in a little bit more in detail. At this juncture, I want to note that you can see on the table there's X's and E's. I point this out every time, almost in every presentation that we do. The E's here mean evaluate. That's to say that even just because you're making a change and that change might impact one of these biological endpoints, it doesn't necessarily mean that testing is the only way to address that endpoint or risk. One way or another, every change has to be evaluated. As you evaluate it, you should evaluate against all the relevant risks. But evaluate doesn't mean test. So that's clear point number one. I know a lot of manufacturers like to just test everything with every change because it feels like they're mitigating some regulatory risk. That might not be the most skillful or best way to do it. Other manufacturers, they never want to test; they only want to evaluate, and that's not always the strongest thing to do either. So we have a set of risks. When we make a change, we want to evaluate against those risks. We want to remember that evaluate means a variety of things to try to understand if those risks are real or not necessarily a test. a big part of this, I would say a big part of medical device biocompatibility as a whole today, and also a really big part of how we can approach changes, goes back to toxicological risk. And you'll see when we go through an example or two how this idea of toxicological risk comes into play with medical device changes. So we should talk about this too. So of those things that were listed on top of table A1, I have some of them listed here, and of those, some are very clearly systemic toxicological effects. So for example, the ones that are shown in green are definitely clearly systemic toxicological effects, where I say systemic tox means adverse events that are related to a chemical substance that can get distributed throughout the body, either by the vascular system or the lymphatic system. Now, sensitization, I would argue all day, is also a systemic toxicological effect, but it's one that has a very local reaction, right? So there's a whole system response, but produces a local reaction is from a testing perspective. So that one's not highlighted in green here. MMP maybe could be considered systemic. Of these systemic toxicological effects, the ones both in green and with red arrows, the FDA has explicitly said, okay, we'll accept chemistry information and a toxicological assessment to evaluate those endpoints. And so if you have a device and you're making a change and those endpoints are on scope, then you have this ability or the option of using chemistry and a toxicological risk assessment for those endpoints. Now, and that's to say, if we think about this in terms of options, and that's a good thing to note and remember too, when approaching biocompatibility in general, and also especially when you're talking about changes to a device and how that might impact biocompatibility, you really do always have options, right? So there's no one way to do this. You have choices. Each choice has pluses and minuses. For these things that are within the realm of systemic toxicities, you very clearly have three options. You often can do a written evaluation to address those without any testing at all. And we'll give an example of that. You still have the ability to do traditional biological tests. I would say, carcinogenicity probably isn't a traditional biological test that you would do, though it still is an option. But we see people doing acute systemic and subacute subchronic tox along with genotox as their traditional biological tests, as opposed to written evaluation or tox assessment. In Europe, the traditional tests are less favored now, more and more all the time. So that's why I got a dashed line. And then, of course, you can do a toxicological risk assessment. That would mean some form of chemical analysis followed by toxicological risk assessment. So if we are going to leverage the field of toxicology or toxicological risk assessment to make a claim about safety due to some change, then we really need to talk about toxicology and toxicological risk assessment at least a little bit. So I'll do that now. I have my one slide intro to toxicology. And again, I do this because it sets up the framework to get us thinking about, what sort of things are in the end going to impact safety and in the end going to impact whether or not we have a serious risk on our hands or not. So here's my one slide intro to toxicology. The whole field of toxicology seeks to answer one very fundamental question. And this is a question that is, as I guess, straightforward as it seems today, is still so very, very often missed. And I would say also missed in the traditional biological tests, right? And that question is, how much of a good thing is too much? And that answer to that should consider the route of exposure of whatever the thing is, the duration of exposure, and what type of potential negative outcomes could be there. So how much of a good thing is too much, considering all of those factors? And so to illustrate this, I like to give a very personal example, using myself as the test subject or animal model, I guess. And I think about, okay, well, what happens to me? What's my dose response for Diet Mountain Dew, right? So if I think about my exposure to caffeine through an acidic aqueous vehicle, oral consumption, what happens to me depending on the dose of caffeine that I have. So you can see there on the x-axis, that's the daily dose in milligrams per kilogram of caffeine. And on the vertical dose is some sort of biological response. So if I have 0 caffeine, then there's a negative response. So I would be irritable, have headaches, generally be just unpleasant to be around to all of my coworkers, that type of thing. And actually to have a negative response when you have no dose of something, this is pretty common in the world of toxicology and biology, right? So you can think of every essential vitamin and mineral that we have. If you have too little of that, then that's a problem. You need to have some. But if you have too much, then vitamins and minerals become poisonous right away. So there's something like 100,000 cases of acute vitamin A poisoning in the US every year. And that's people just taking too much vitamin A. So you need some, but too much is a problem. Okay, so if I have more caffeine, to where my new normal is, then that's the minimum of adverse effects. That's when I'm feeling great, just riding this crested wave of caffeine in my system. But you go higher and higher, and you start to see adverse effects coming into play. So eventually you get some arrhythmia, tremors, you get all shaky, hallucinations come in at a certain dose. And then you reach this point where, depending on what you're measuring, you have a 50% chance of something bad happening. So there's some point where if the bad thing happening is death, there's a 50% chance of death, and that would be the LD 50 or the lethal dose 50. And then you always have a maximum response where, using the morbid case of the adverse effect being death, would be that every animal in an animal model dies as a result of an exposure that large. And for caffeine, that's something like 150 milligrams per kilogram. So that much caffeine is guaranteed to cause death really in any animal or a human, right? And I calculated it. I think that's about 95 Red Bulls. So that's, it's quite a lot. So this is the whole idea of toxicology, understanding dose to response, especially when we think about dose to some sort of adverse response. And more often than having some personal homeostasis, the data exists out there as doses that were exposed to some animal system, mice or rats or rabbits, and then what happened as the animals were given higher and higher amounts. And so you can see usually the data exists like this S-curve that you see on the screen here, where there's some dose where it's the highest dose where nothing bad happened. This is the no observed adverse effect level. And then there should be one dose higher, the lowest observed adverse effect level. And that's kind of an important data point, because it shows that the animal model and the entire test system was capable of measuring an adverse effect, right? And so this data exists out there in the literature, and the whole art of toxicological risk assessment then is about comparing, what might be coming off of a device, let's say as measured using some chemical analysis or even some theoretical amount that might come off of a device. It would be comparing those amounts with this type of information that's in the literature. So it's let's say that we see cyclohexanone coming out of some soft plastic device. But the amount that comes out is, several thousand times less than a no observed adverse effect level that's out in the literature. Then we can say, well, okay, we have a mechanism to compare the amount that was found or the amount that might be there with something that we know is not going to cause any adverse effects. And we use these toxicological ideas in evaluating medical device changes. Now, another idea that's really important to both toxicology and to the evaluation of changes is this threshold of toxicological concern. And it's worthwhile to take a second and talk through this at least a little bit. So the threshold of toxicological concern is, what if you have some chemical, but there's no literature out there, it's never been tested before, and so you really don't know how toxic it might be? Or what if you have a chemical, but you don't know very perfectly like what that chemical is? You know it's there, but you can't say what it is 100% for sure. Well, this problem has existed for a long time. It's existed since the 60s and 70s, when people really started to use chemistry to talk about safety. Back in the day, it was mostly applied to food levels, right? So how much of an unknown thing is okay in food, or how much of a pesticide is okay in food, and that's where it comes from. And so the threshold of toxicological concern is the amount that if the exposure is below this amount, even if the chemical is unknown, we don't care about its toxicity because we know it's going to be safe. Or even if we know that it's a potent carcinogen, if it's below that exposure, we really believe that it's going to be safe too. And so this really helps when you have, let's say, a very small change to a device, and you don't know what sort of chemicals that change could make. So for example, think about something like a very, very small sensor wire. So sometimes we have sensor wires where the wire itself weighs only a few milligrams total. And you make a process change to that sensor wire that's upstream. And because of this process change, let's say the MSDS for a new chemical is only 97% pure. So that's like 3% of that new chemical is unknown. And you could compute, well, okay, how much of that 3% might actually end up on this sensor wire? And it's like a picogram. Well, then, a picogram, even though you don't know what it is, that's definitely going to be less than a threshold of toxicological concern. And that's why we wouldn't want to go through this whole rigorous process of testing and analyzing for that if we know it's going to be below TTC anyways. So we use this idea of TTC pretty rigorously, and it's a very powerful tool to use. One that I would say has been, in general, very convincing to regulators, both the FDA and on a more global scale. Okay, so we have to have this TTC idea out there because we use it all the time. So with that, I know I've been talking pretty fast. Then let's just go into some case studies, and I'll talk through it, and we'll see, how some of these ideas come into play. So there are a couple that have been submitted into the chat. So let's take this first one, right? So the question is, could you talk about a CPAP tubing and heated humidifiers? Well, the asker of this question has hit on a very hot button topic where there's been a lot of, let me say, grief with the FDA. So the FDA has more recent years had some issues with CPAPs, and they've been highly scrutinizing this class of device. So we have to say that we're talking within that framework as well. But when we talk about CPAPs and heated humidifiers, there's the first thing that we want to think about is what's the nature of exposure, duration of exposure, and all that. So a CPAP tubing, if we're not talking about the mask, just the tubing, then it only contacts the patient in an externally communicated way through the gas into the patient's lungs, right? And this type of contact squarely puts it in the realm of ISO 18562. So ISO 18562 is the standard set that applies to respiratory contact medical devices. If the device is humidified, which in this case it says heated humidifiers, then that means that the testing regime for this type of device is going to be pretty elaborate. So what we would do is we would evaluate inhaled VOCs, inhaled particulates, and then because regulators are worried about condensation inside the tubing set that can then be swallowed by the user, there would be an extractables chemistry program, followed by a toxicological risk assessment of those chemicals that are extracted. Now, in the context of a change, right? So let's say that we have a CPAP and a heated humidifier that is now undergoing a change, and the existing CPAP tubing and heated humidifier are fully cleared and on the market. Then what we would do is we would ask ourselves, how could that change potentially impact any biological endpoint that's in scope for the device itself? And for something like a CPAP tube, really most changes could one way or another impact the biocompatibility. So for example, if the change is the packaging, so let's say that the CPAP tube is going to go into a different plastic bag. Well, CPAP tubes sit in that plastic bag for a long time, and any volatile organic chemicals from the bag that could be there, they have plenty of time to slowly migrate into the CPAP tube materials as well. So if you were to change a plastic bag, we would worry or think about transfer of volatiles from the bag to the CPAP tube. And we would want to check, okay, could those, are there new VOCs that are being transferred? And if so, how much, right? So that's the type of thing that might produce a VOC test. If we're talking about the change of a supplier of polymer in a CPAP tube, let's say they're both PVCs, though the PVC is definitely being phased out for CPAPs. Let's say that they're both PVCs, one from supplier A and the other from supplier B, and we don't know if the upstream suppliers are the same. Then that would be the type of change that really would be difficult to evaluate without a full suite of testing, because the FDA knows and we know that making a change from one PVC supplier to another means that the formulation of the PVC could be different, and the extractable compounds from that could be different as well. Sometimes, I'll talk about one more type of change that we see with CPAP tubing. Sometimes the only thing that's changing is the proportion of different materials. Now that becomes a really easy one. So let's say that there's two material types, a connector type and the rest of the tube, and now they're making the connector a centimeter longer and the tube a centimeter shorter. Well, that's a case where there's really no new materials coming into the system. So there shouldn't be any new volatiles. There shouldn't be any new anything. It's just the relative proportion of those. And for something like that, if the change is relatively small, where small means less than plus or minus 10%, or depending on the nature of the change, less than plus or minus 30%, then we would say, yeah, that change is too small to even theoretically produce a different test result if we were to be tested again. So small changes that don't introduce anything new, that could be something that can pen and paper be handled pretty quickly. So there's a lot of possibilities with CPAP tubing and heated humidifiers. If the asking person wants to like narrow in more specifically, I'd be happy to go more specific. But that generally is it. Okay, so there's another question about changes. It says, are we required to evaluate biocompatibility for a device located out of the sterile field? If yes, which 10993 standards would apply? And so the short answer there is yes, right? So the need to evaluate biocompatibility for a medical device really boils down to not necessarily if it's within the sterile field or not, but it's more about how do those devices, so what's claimed for the device, how is it going to be marketed, and how does it contact the patient, right? So there's many devices that, let's say, that are at-home medical devices that are marketed that way, not sterile, that definitely do have the full suite of biocompatibility on them. So I think about things, for example, like a CPAP or an oxygen concentrating machine, infrared thermometer, for measuring your child's temperature. All of those things are medical devices that undergo biocompatibility evaluation. So So I guess the short answer there is that yes, those are something that would be needing biocompatibility. Okay. So I'll keep an eye out if there's any more specific types of questions that want to be talked about, and I'll go into the one that's on your screen here. So this is a real change that we handled recently. I kind of anonymized it a little bit, but most of the detail is really exactly the same. So we have a catheter that is manufactured with a process that includes several dip coating steps. So anybody that's in the catheter business knows that typically there's a lot of dip coating. And then in one series of these steps, a portion of the device is masked by polyvinyl acetate. The device is dip coated in a way that the new coating overlaps with the mask, and then later the PVA mask is removed to give a nice crisp line between the coated and the non-coated parts, right? So what they wanted to do, or the nature of the change is that they, through some additional engineering and experimentation, they found that actually they could produce this crisp line between the two layers without needing the masking step in the first place. So the change in this case was to simply remove the PVA masking step, and all else within the entire manufacturing process flow would remain exactly the same. So this is one that hit our desk. So when I think about this, the manufacturer is asking, okay, so should we retest everything? And my quick answer was no. I don't feel like they should retest everything. In fact, I don't know if they needed to do any testing, right? And the reason why is, like, if I go back to that scope of biological risks and think about what's really happening here with this change, then what I see is that really the only thing that's happening is they're removing a set of contact chemicals in this process. So there was PVA and all of like the solvents and impurities that might go into that PVA masking step. That was there. All of those are no longer there. So really, at least on the surface, there's no increased risk. Actually, there's a decreased risk. And you think about it in terms of like cytosensitization, irritation, acute systemic, and through the list. And really, there's no way that removing this a set of contact chemicals could be adding risk. So overall, it says, okay, it looks like we're really decreasing risk. Their question was, and this is a very conservative and thoughtful manufacturer. Their question was, well, how do we know that adding the PVA didn't like cure something or somehow block an adverse response that would have been there? To look at that, we looked at we had an expert look at the chemistry of the materials of the device and also the chemistry inside the PVA mask, and we could see there's nothing there that was reactive at all between the two materials. So we could say, it's fair question. What if the PVA was like reacting with something to lower the risk, but there was no mechanism for that? So in this case, the approach for evaluation was to say, okay, here's the device. Here are the biological endpoints required for evaluation for that device. Here's the change. And because the change is like this, that means none of those biological endpoints could be negatively impacted. So there's still a written evaluation. It still goes against those endpoints, but it's just explained why those aren't, why the change doesn't impact any of those risks. Okay, so similarly, same type of device. It's different class, same manufacturer. So, for one of the coatings discussed with the PVA masking step, the material to be deposited was dissolved in a solvent. So we had the PVA mask. They were going to dip coat it. As they dip coat it, that's a process where you have something that you intend to leave behind that's dissolved in a solvent. And the supplier of the solvent was going to change, right? So they were going from supplier A for the solvent, let's just say the solvent was ethanol, right? So supplier A for ethanol to supplier B for ethanol. And as we looked at this change, we thought, okay, so the main ingredient is the same, ethanol. But the nature of the manufacturing process really is to use ethanol as just the matrix to support the thing that they're depositing, and then the ethanol is really intended to just completely evaporate away. So in the final finished form of the device, there really should be no ethanol at all. But then the next question was, well, what about the impurity profile of the ethanol? So let's say supplier A and supplier B have different ethanol impurities in place, and we don't know what the impurities are in the new ethanol, and we know that some ethanol impurities could be non-volatile, and so they might be left behind on the device, right? And so here's where the TTC came in, right? So we were able to calculate, as the catheter comes out of the dip coat, how much ethanol might still be on it as the catheter is drying. And then, based off of that amount of ethanol that could be on there as it was drying and the purity of the ethanol, so, most ethanol is 95% because that's as pure as you can get by distillation. It's 95% with water and a very, very small amount of other impurities, so like 0.1%. So we calculated the amount of ethanol on the outside of the catheter as it comes out of the dip coat and use that percentage of the purity to show, hey, wait a second, even if there are impurities, new impurities from the ethanol, based off of that percentage, we know that those impurities could not be above the TTC. So to evaluate this, we explained what the catheter is, the biological endpoints, and then we showed this calculation where we say, okay, yes, we've thought about this, and it's not really possible for any impurity to remain above the threshold of toxicological concern. Okay, there's another question that came in, another example. So one is, how would you advise handling using of two types of FEP liners that are consumables on a catheter? The liners only contact the catheter during molding, is not part of the catheter after that. Yeah, so this is like any of these other things where something contacts a device during manufacture, but it's not part of the final finished form of the device. And there, the real question is during this contact. So when the device contacts, let's just say, this manufacturing fixture, I'll look at it that way. Can there be anything that transfers from the liner to the device? And we'll often look at that and say, okay, how long does the device contact the liner? And how much time is really needed to have effective transfer of something from something like an FEP liner to a catheter? And more often than not, there's some sort of evidence or supporting information that the manufacturing fixture or the liner or whatever is clean, right? So if the liner's clean, that means that there's some evidence that there's not residual materials on the surface of that. And if the device if the fixture is clean and it contacts the device, then there's really very little chance for transfer of unwanted chemicals or materials from the liner to the catheter. And so what we would generally do is go through and explain that using some supporting information like evidence that this thing is clean. Now, if there's not evidence that a manufacturing fixture is clean, then in that case, we would look for that evidence. So we would say, okay, can we measure those liners and prove that they're clean? Or can we look at catheters that have been in contact with both liner types and just show that there's not a difference in residuals between the two liners? And the good thing about those types of questions is that there's usually analytical methods that are compared to the suite of biocompatibility, fast and very effective and inexpensive that could be used to answer that question. So we might, for example, run an NVR and a TOC on both liner types and show like, yeah, there's no difference in NVR and TOC. That means that they couldn't be transferring anything different by an appreciable amount to the catheters, and they could be considered equivalent. So there's ways to approach it where it boils down to what could transfer from the fixture to the device, and how can we know that? How can we show that that amount isn't going to be significant or concerning? All right, let's do one more example, and then I think that we'll probably have time to open it up for questions. And so this is kind of an interesting one. So selective laser melting or selective laser sintering, there's probably another name for that process too, is a process where you deposit very thin layers of some powder. It could be either a polymer powder or a metal or even ceramic. And you melt a thin layer of that together in a certain shape. So it's a type of 3D printing or additive manufacturing. And this has been commonly used now for, gosh, I want to say at least 5 years in orthopedics. It's pretty easy and effective to manufacture titanium and other metal implants this way. And we've seen more and more where this is applied to polymers, right? So in this particular case, we have a permanent implant device that's selectively laser melted from a polymer powder. It's built up layer by layer. And then finally at the end, that powder is blown out and cleaned out, and this intricate 3D device remains. And in this particular case, the powder that was deposited is a mixture of the polymer and a small amount of flow agent, a biocompatible, well-known biocompatible flow agent. Let's put it that way. And what was happening was that the manufacturer was buying the powder and also buying the flow agent, and they were mixing it themselves. And then that mix would be going into the printing process. Well, what they want to do, or the nature of the change was rather than them buying the polymer and the flow agent and mixing it themselves, the supplier of the polymer could also buy that exact same flow agent, and the supplier would just mix it for them and give it to them. So it removed the responsibility to mix the powder and the flow agent from the manufacturer just to their supplier. So that was the question that was being asked. And so when we think about this, right? So the bulk materials here remain identical. So this is just a processing change question. And even then, the process itself remains identical. It's really more the location that the process is taking place. So rather than, the mixer at this manufacturer is going to be the mixer at their supplier, but the supplier buys the same mixer even. And so when we started to look at evaluating this, we looked at, okay, well, so really the only way that there can be increased risk is if the supplier or the location of the new place for the mixing, if they don't keep their mixer clean to the same way that the manufacturer would, or if the mixer is in an environment that could introduce contamination or other unwanted manufacturing materials. And so that's how we evaluated this one, is we said, okay, the existing mixing process happens, let's say, in a certain type of clean, controlled environment, not necessarily a clean room, more like a gray room. And the mixer itself is only used for this purpose, and it's maintained this way. It has a certain cleaning process at regular intervals. Does the manufacturer, the supplier, have the same process in place, even though they're in a different country, different building, all of that stuff? And by looking at that, we could say, actually, yes. So the processor, the supplier is using the same manufacturer, same mixer, agrees to also put it into this same type of clean, controlled environment, and will buy the same cleaning materials to maintain this mixing machine over time. And then in that case, it's really parity, right? So there's no increase or new risk, provided that the environment and cleaning process for the machine are the same. And then, all of this is written down in a very nice, articulate biological risk assessment that evaluates this process change. Okay, so with that, I think I'll go to straight up Q&A, and I'll turn it back to the host to address those questions or ask those questions.
Well, here we go. Thank you, Dr. Do.
My pleasure.
Okay, there you go. Dr. Do no harm. Okay, so let's go to our first question. Okay, should it be called? Oh, well, actually, let me intro for anyone who is not sure how to use the function. Before we begin, also a quick survey. Before we do that, direct your attention to our quick webinar survey available on the right hand of your presentation window. If you close the survey, you can reopen the widget by clicking the icon at the bottom of your screen. And thank you in advance for filling out any feedback on the form because your participation really does help us serve you better. Now, onto the Q&A. As a reminder to participate in it, just type your question into the text box located to the right of your presentation window, or alternately, you can click the Q&A icon at the bottom of your screen. If we're not able to answer all submitted questions during today's webinar, we will be sure to share them with Matthew, who can reply to you offline. Now let's get to it. Should it be called biological safety evaluation or biocompatibility risk assessment? And are there any differences?
That is a good question. We say, so when I say we, Nielsen Labs, as we provide this as like a consulting service, we say biological evaluation report as a summary document, or we say biological risk assessment if we're talking about a change. I see more commonly used than the words that we're using, biological safety evaluation or BSE. In our experience, the precise name of that document hasn't been super important to regulators. What's more important is that these documents are written with a careful eye towards the audience, right? So you really have to, like, you would think that these regulators, the people reading these documents, are experts, that they have a good background, let's say in chemistry, anatomy, biology, and all of that. But you really have to write the document with a clear understanding that actually most of these regulators or reviewers aren't experts. Probably they've never taken an anatomy class, or they maybe have never even taken a chemistry class, right? So more than the title of the document, it's really important to write it clearly so that a non-expert can understand and still have enough detail so that it can survive expert scrutiny. I hope that helps to answer the question.
Yeah, I think so. Very good. The next question. What about instances when changes to suppliers occur? For instance, a supplier of gloves used to handle a device in production in a control room prior to radiation sterilization.
Yeah, that's a great question. So in general, we think about things like that, right? So let's say a change in gloves as being really, really low risk. However, every change, including that type of change, really must be evaluated. And when you've been dealing with medical devices long enough, you start to see changes like that actually have an impact on biocompatibility. A classic story is a case where there was a glass fiber device, and that exact thing happened. The manufacturer changed glove types from a nitrile to a powdered latex glove and started to fail cytotoxicity because of that change, right? Or had a difference in result of cytotoxicity as a result of that change. So really anything that contacts the device, including gloves, if that changes, it should be evaluated one way or another. And as you approach that evaluation process, you have to put your skeptical regulator hat on and ask some tough questions. Could anything change the device? And in the case of gloves, yeah, it could, because if there's a difference in, let's say, residuals on those gloves and those gloves touch the device, then that means that the residuals on the device could be different.
Very good. Let's go on to the next question. Does the LD50 and max response have a particular time constraint associated with the dosing? Secondarily, how does that time aspect impact evaluation of risk, if at all?
I think that that's a great question, and it's one that's worth digging into at least a little bit. So LD50s, this is the lethal dose 50, where they've given progressively higher and higher doses of a chemical to an animal system. Usually it's in rats, where at least half of the animals died. And this is a death, in this case, is an acute response, right? So it's not that they died slowly of cancer, it's that they died of an acute poisoning. So that's, something to keep in mind. That's what's being measured there, is an acute response that produces death. The other thing about LD50s is that they're really highly accessible, right? So this is a very commonly measured metric of toxicity, and it's also accessible because it's often included in safety data sheets, and we all have to have safety data sheets for all the chemicals that we have on site in a manufacturing facility, so it's like extra available. So it's not recommended. So from a general tox perspective and from a regulatory perspective, it's really not recommended to use LD50s to make some sort of safety claim at all, right? So we don't want to use death as the endpoint to compare something that we're, when we're making a device that's trying to help somebody. So it's not like the right thought process anyways. And then the other thing is that, since it's an acute response, and a lot of the times if we're doing chemistry and a toxicological risk assessment, we're looking at repeated doses from slow leaching of chemicals out of a device. It's not super fair to compare an acute response to a really long duration, repeated dose exposure type of interaction. But all of that said, there's, sometimes you do everything you can to avoid basing a conclusion on an LD50, but there's nothing else out there. So let's say there's no tox information out there at all. The only data point you have is an LD50. So when you find yourself in that situation, then you can apply some uncertainty factors to extrapolate from an LD50 to something that you feel confident, applying to human safety. And the general consensus there is that you should have an uncertainty factor of 10 for interspecies variation, another factor of 10 for intraspecies variation, and then a factor of 100 for the data quality. And that's because we're talking about, death to a no observed adverse effect, and also acute to a chronic situation. So those factors together, 10, 10, and 100, make a cumulative factor of 10,000, right, which is very, very conservative. So I would say avoid LD50s altogether. If you are backed in the corner and you have to use an LD50 because there's nothing else, then it's generally well accepted to document that you've done your diligence looking and to apply this factor of 10,000 in deriving a tolerable intake or exposure.
Thanks. Now this is about plans and reports. The details matter. So here's a question. Do we need a biological risk assessment to complement a BEP and a BER? Always.
Yeah.
I guess I emphasize always,
Yeah. I think that it's really important to try to remember how these documents are going to function for you in your whole system and submission path. So I would say a biological evaluation plan in one form or another is always required. I would never submit to any regulator without them knowing that I have a plan. And that plan means, what is the device? What are the risks? What are we going to do to address those risks? So that's the function of the BEP. I say it's required. Even if you call it by a different name, still that's required. The BER, the Biological Evaluation Report, or the Biological Safety Evaluation, depending on how you call it, this is the summary document that writes in plain English for a non-expert and an expert that we did everything that we needed to do to evaluate the safety of the device, and it is in fact safe because of X, Y, and Z, right? that document is required. I can't see a successful submission without that in one form or another. Again, there's a whole art to that. I highly recommend that you write that in a certain way to make it accessible. So those are the two capstone documents. Now, a biological safety assessment. This could be used to address a certain type of risk, and it might be referenced in the BEP or the BER, but it's not always required, right? So it could be that the BER says, yeah, we were worried about the use of gloves, and so we did a biological risk assessment, and it was okay, right? So, and then if the reviewer wants to know, okay, well, how do they really know that these gloves aren't an issue? They can go to that biological risk assessment and see that. So I would say that a BRA or a risk assessment in that form isn't always required. So that is not always required. But it could be, just in case that, in case you use that type of evaluation, let's say a pen and paper evaluation to address some sort of risk. So I hope that helps.
Okay, very good. Is there a reference for the safety factor of 10,000 that you mentioned just previously?
Yeah, there is. So, maybe the best way to communicate that, isn't by me chatting over here, but I would say we can follow up. With a link to some of that. There's really two angles there. One is the more informal discussion in 10993-17 on the application of uncertainty factors, and another is an academic article that really defends the use of that 10,000. So.
Okay. We are just about up against time, so maybe the safe thing to do to make sure we don't get cut off, we risked it yesterday on day 2, is to say that's all the time we have for questions today. But there are some more, and as we said before, they'll be submitted to you offline to handle them. Thank you, Matt. We appreciate your time and expertise on the topic today, and thank you to Nelson Labs, as well as to everyone in the audience. We appreciate your attention and participation. Within the next few hours, you'll receive a personalized follow-up email with details and a link to today's presentation on demand. Please feel free to invite your colleagues and peers who may not have been available to listen to the event today, as well as day 1 and 2. And if you missed any of the classes, as I said, from this 3-day Focus on Fundamentals series, be sure to click the Register Now button in your viewing window, and then all sessions will be available to access them on demand. This webinar is copyright 2022 by Informa. The presentation materials are owned by or copyrighted by Informa Markets, and the individual speaker today and all days are solely responsible for their content and opinions. And on behalf of our guest, Matthew, I am Bob Sperber. Thanks for your time and have a great day.